And most of us think about the bottom of the food chain. We think of something like grass or trees, plants that use the sun's energy to provide food for higher organisms. But what starts the flow of energy in the ocean? Plants have roots, and so they can only grow so deep. Phytoplankton are the microscopic floating organisms responsible for nearly all the primary production in the ocean and half of the primary production on Earth. This diverse group of organisms come in all shapes and sizes, and like land plants, some are indicative of a healthy environment, while others are toxic. Studying phytoplankton is a great way to get at the health of marine ecosystems. And while you can use this to study ecosystems anywhere in the world, I wanted to study those right here in Charleston Harbor. As you've seen, episodic storm events, like pointed out before, have wreaked havoc in our area and caused issues like increased flooding, stormwater runoff, and even blooms and toxic algae. So what makes studying phytoplankton so important? Those things. So I want to look at what's happening right here in our harbor right now, and also hope to generate a baseline study for changes in the future. So studying phytoplankton can be a bit complex. There are a bunch of different methods, and none of them are perfect. Each method has different advantages and limitations and gets at a different aspect of the community. It's actually recommended that we use a variety of methods to get the best idea of what's going on in our system. I'm going to use 3 methods. The first method is microscopy. This is just looking at ourselves under a microscope. It's pretty straightforward, but takes a lot of patience and a lot of practice. Next is something called HPLC pigments. This separates phytoplankton based on the different pigments they use in photosynthesis. Finally, we have DNA metabarcoding. This identifies phytoplankton based on the unique DNA sequences found in the cells. So the 2 objectives for my study, 1 are to see what phytoplankton we have here in Charleston Harbor, and 2 to compare and contrast these different methods. I've been able to see some results already, so what we can see is that different groups of phytoplankton do dominate at different points in our season, and we are seeing many groups coexisting at the same time. I have looked a bit into my methods as well, and what it seems is that HPLC and metabarcoding are showing similar results, while microscopy is delivering different information. I'm excited to explore the rest of my results and see more into the diverse plant communities that make up